In September 2025, a team at the Max Planck Institute for Evolutionary Anthropology in Germany published the most comprehensive ancient DNA study of Slavic origins ever attempted. Dr. Joscha Gretzinger and Dr. Johannes Krause, two of the most respected names in ancient genomics, led a consortium that sequenced 555 ancient genomes from burial sites across Poland, Germany, Croatia, and Ukraine. They called it the HistoGenes project. And the number they found for Poland was not a surprise. It was a detonation. More than 80% of the people living in what is now Poland, the people who built the settlements, worked the fields, buried their dead along the Vistula, were replaced. Not gradually, not through slow mixing over centuries. Replaced. Within roughly one human lifetime, when the genetic identity of an entire region was overwritten by people who came from somewhere else entirely. Stay with me, because that number is the reason this video exists. Think about what 80% actually means. It means that if you lined up 10 people in that Warsaw classroom, 10 people who believed their ancestry was ancient, local, and unbroken, 8 of them would descend in their male lineage almost entirely from migrants who arrived from what is now southern Belarus and central Ukraine around the year 550 CE, not the Vistula Basin, not the Polish forest, the Eastern Steppe Corridor, and the people who were there before. We know something about them. At Kołalewko, a burial ground in central Poland that was active from roughly the 1st to 3rd century CE, a Polish research team recovered Y-chromosome data from 16 male burials. 8 of them carried a haplogroup called I-1, a lineage genomically adjacent to Iron Age Scandinavia. These men buried Roman bronze vessels, Baltic amber beads, and locally carved bone combs. They traded across a continent. They had an organized community. They had a world. They were not Slavs. And within 2 centuries of the Slavic arrival, their genetic lineage was almost gone from Polish soil. If you've ever wondered where you really come from, not what the textbook says, but what the bones say, subscribe, because this is only the beginning. The Slavic migration is supposed to have been slow, organic, generational, but the genomic compression in Gretzinger's data tells a different story. The replacement of a region's entire male lineage appears to have happened within a window so narrow, the HistoGenes researchers flagged it specifically in their supplementary discussion. How fast was it exactly? We'll come back to that number. And when we do, it won't feel like history. It will feel like something that happened to a person. Before we go any further, I want to know something. Have you ever taken a DNA ancestry test? And if you did the results match what your family always told you? Drop your answer in the comments. Poland, Pakistan, Nigeria, Mexico, wherever you're from. Because by the end of this video, I'll show you why that gap between family story and DNA result is the most important thing in modern genetics right now. A person. Seen, not a culture, not a type. A specific man. Kołalewko, Central Poland, approximately 150 CE. His Y chromosome, I-1-L1237. His grave, a Roman bronze vessel, a bone comb carved by hand, 3 amber beads from the Baltic coast, 300 kilometers north of where he was buried. He traveled, he traded, he was part of something real. And his culture has a name that he never knew and never used, because the name Wielbark comes from the village where his graves were found in the 19th century. Whatever he called himself is gone. Whatever language he spoke is gone. His genetic lineage would survive in modern Poland in fewer than 7% of men alive today. Dr. Ireneusz Stolarek and his team at the Institute of Human Genetics at the Polish Academy of Sciences spent years building what became one of the most important datasets in Central European archaeogenomics. In 2023, they published the results of analyzing nearly 500 individuals from 27 Polish cemeteries, 197 of them with full genome-wide data. What they found was that Wielbark culture individuals, the people who built Kołalewko, who buried the man with the amber beads, showed genetic profiles closest not to any Slavic population, but to contemporary Iron Age Scandinavia. These weren't proto-Slavs waiting to be replaced. They were a distinct people, genetically specific, who had been building cemeteries in Poland for 400 years. And yet, the data said something that nobody in the popular press has picked up, something that changes the entire frame. The Wielbark people didn't just disappear when the Slavs arrived, Stolaric's team found that when you try to model the genetic makeup of the medieval Slavic population that replaced them, using the mathematical tools of population genomics, you can't do it cleanly without including the Wielbark signal as an input. The replacers needed the replaced to exist. The people who supposedly vanished left a trace so embedded in the Slavic genetic profile that without them, the math doesn't work. They are mathematically invisible and statistically present at the same time. At Masłomiec, another Wielbark site in eastern Poland, Stolaric's team sampled 27 individuals from the 2nd to 4th centuries CE. Of one specific woman buried there carried U5b mitochondrial DNA, a lineage linked to the Western hunter-gatherers, the people who lived in Europe before farming arrived, before the Bronze Age, before any of the populations we've talked about. She was a Wielbark woman carrying a Mesolithic ghost inside her cells, the oldest thread and the newest one in the same body. The Wielbark are not a footnote. They are the chapter that got torn out. And nobody asks about them at a popular level, not because the data is hidden, but because what the data shows is so rapid and so complete that explaining it means asking the one question Polish archaeology has been stepping around for decades. Not where did they go? Why did the men leave? And not the women. There is a migration wave in the record, somewhere between 450 and 550 CE, that swept the male lineage of northern Poland to near zero within 100 years. Two small things survived that sweep, and the first one is the most unexpected: the women. That's where the answer starts. But before we get there, hold everything you just heard, because it's about to mean something completely different. Every account of the Slavic expansion into Poland tells the same story. Vigorous eastern people moving into available land, filling a vacuum. The Wielbark move west with Gothic migrations. The old cultures fragment. The land empties. Then the Slavs fill it. Clean, directional, simple. Except Gretzinger and Krause's 2025 data won't let that stay simple, because the Wielbark male lineages were already fragmenting and contracting before the confirmed Slavic arrival window of 550 CE. The pre-Slavic populations hadn't been displaced by Slavs. They had already begun to leave. So who moved them? The Goths. This is where it gets strange. And I mean genuinely strange. Ancient DNA now confirms what archaeology had long suspected. The Goths, the people who sacked Rome, who founded kingdoms in Spain and Italy, who are among the most documented barbarian peoples in history, grew directly out of the Wielbark culture of northern Poland. Stolaric's 2023 study found that Wielbark individuals carried the same Y-chromosome haplogroup composition, I1 and R1b, that reappears in early Gothic individuals sampled in southeastern Europe. The people who ended the Western Roman Empire walked out of the Vistula basin. They were the sons of the man with the amber beads, and when they left, they took the fathers with them. The Gothic departure wasn't a single event. It was a generational drain, running from roughly 200 to 400 CE, as Wielbark men followed what historians call the Gothic Road southeast toward the Black Sea. Each generation moved further south. Each generation left fewer sons behind in Poland. By the time Slavic migrants arrived at Grodek on the Bug River around 550 CE, they may not have been conquering occupied territory. Whilst they may have been walking into a wound that the Goths had already opened, which means the Slavic ancestors of modern Poles were not conquerors in the way the story is usually told. They moved into land that had already been hollowed out. And a wound filling is not the same story as a conquest. It changes the emotional weight of that 80% replacement completely. But the data hasn't accounted for everyone yet. The Goths triggered the vacuum. The Slavs filled it. That's the corrected version. Except there are still the women who were in Poland during the Gothic departure, during the Slavic arrival, during that entire century of replacement. And the 2014 ancient DNA study from this region found something in their mitochondrial genomes that neither the Gothic theory nor the Slavic theory fully explains. The women didn't move, not all of them. Some stayed. And what they left behind in the genetic record is the closest thing this science has to a ghost story. This isn't ancient history. This is in Polish DNA right now. Dr. Mateusz Wrobel and his colleagues at the Institute of Human Genetics at the Polish Academy of Sciences published a study in 2014 that most people interested in European genetics have never heard of. The team built mitochondrial DNA profiles for 23 individuals dated to the Roman Iron Age from Wielbark and Przeworsk culture sites in Poland, and 20 individuals dated to the medieval period between the year 1000 and 1400. They were looking for evidence of how the Slavic migration changed the maternal line. What they found broke the model. So two mitochondrial haplogroups, H501 and N101A2, appear in the Roman Iron Age samples. They appear again in the medieval samples, and they are present in modern Poles today. The same maternal lines, continuous, unbroken. Across the entire window of the 80% Slavic replacement, some women's maternal lines just kept going. The paternal genome of Poland, the Y-chromosome story, was replaced. The male haplogroups of the Wielbark culture, I1 and R1b, effectively vanished from Polish soil by the 8th century CE. But the maternal line didn't break. A subset of women survived the Gothic departure, survived the Slavic arrival, survived a century of demographic upheaval. And their daughters survived, and their daughters' daughters are alive in Poland right now. There is a woman buried in a Wielbark cemetery near the Vistula River, sometime around 300 CE. Her mitochondrial haplogroup is H501. Her name is not recorded, her language is not recorded, her face is not recorded. But in a Warsaw laboratory, during the analysis for the 2014 study, a researcher identified the same sub-clade in the genome of a modern Polish individual. Not similar, the same molecular signature. Approximately 1,700 years of unbroken maternal descent. No monument, no ceremony. Just a sequence match on a screen at 2 in the afternoon. Actually, real quick, if you caught my last video about Jewish DNA and what the genetic record really says about Jewish origins, you already saw the first half of this pattern. The same dynamic appears there. Women's maternal lines surviving a mass demographic displacement that almost completely erased the male lineage. It's the same genomic signature in a different population in a different century. Did that video change how you see ancestry? Tell me in the comments, and then stay, because what's coming next is the part that video was building toward all along. Polish genetic identity is not a simple replacement story. It is a story of who survived the replacement, and it was the women. Which raises a question so uncomfortable that the 2014 study authors addressed it in 3 careful sentences in the discussion section, and then moved on. Why did the women stay when the men left? Was it choice, social structure, or something that was done to them? The mitochondrial data cannot answer that, but the bone chemistry at Kołalewko can. And what it suggests about the women of pre-Slavic Poland is not a story of resilience. It is a story of immobility. When the line between those two things is not always a line at all. Immobility isn't the same as survival. The Kołalewko burial ground contains over 300 graves. They are organized, careful, a community that grieved with structure. But inside the bone chemistry of those burials, in a type of analysis called strontium isotope testing, which measures where a person was physically born based on the minerals in their teeth, there is a pattern that the site reports document clearly, and that popular summaries of Polish prehistory almost entirely ignore. Stolaryk's team ran strontium analysis on Kołalewko individuals. What they found was this. A significant share of the women buried at Kołalewko had strontium ratios that didn't match the local geology. They weren't from there. They came from somewhere else. Meanwhile, the men at Kołalewko showed local strontium signatures. The men were born there. The women were not. But here's where it gets strange, because the direction of that pattern has a specific name in anthropology, and it changes everything about who these women were. Patrilical exogamy. Men stay. Women move at marriage, brought from other communities, sometimes distant ones. It is documented across Eurasian prehistory in hundreds of sites. At its most voluntary, it is cultural exchange. At its most coercive, it is something that has a different name entirely. And the strontium data at Kołalewko cannot tell you which one it was. That silence isn't scientific neutrality. It is a genuine gap in what the evidence can say. One specific female burial at Kołalewko, documented in the site records, dated approximately 350 to 400 CE, contained a woman in her late 20s. Her strontium ratios pointed to an origin in the lower Vistula, roughly 400 kilometers northeast of where she was buried. She was not from here. She had traveled, or been brought a significant distance. She carried an amber bead, a bronze ring, a bone comb. She ended here. Her maternal haplogroup connects her to the Baltic fringe, the eastern edge of Europe. She is one of the women in the 2014 study. She is part of the reason H501 is in modern Poland today. Think about that for a second. The unbroken maternal line that survives in Polish women, the one that the genomics community points to as evidence of ancient continuity, may not be a story of women choosing to stay. It may be a story of women who could not leave. The women whose mobility was controlled by the same patrilocal system that moved them to Kołowko in the first place. Constraint, rewritten as heritage. That doesn't erase the continuity, but it changes what we're looking at when we celebrate it. And then, the strontium data reveals one more thing. If the pre-Slavic women at Kołowko were brought from Baltic fringe communities, and the Slavic men who replaced their fathers came from the east, then the maternal haplogroups that survived in modern Poles are not purely Wielbark. They may be carrying Baltic ancestry that predates the Wielbark culture entirely. And to know whether that's true, you have to go all the way back through every layer. The Baltic thread is older than you think. But before we pull it, let me give you the full picture. The complete genomic architecture of modern Polish ancestry, laid out as clearly as the current science allows. Consider this the answer you've been waiting for. There are 3 genomic layers that built modern Poland. The third one is the one that matters, and it's the one no textbook includes. Floor 1. The oldest signal in Polish territory comes from Western hunter-gatherers and early European farmers, people who were living in Central Europe between 9,000 and 4,000 years ago. Dr. Wolfgang Hack at the Max Planck Institute in Germany has spent decades mapping this layer across the continent. In modern Poles, it contributes roughly 20 to 30% of total ancestry. These were the Ice Age survivors and the first farmers, people who carried the U5 mtDNA lineages you see at Kołowko. You're people whose blue-eye alleles are still visible in Polish faces today. Floor 2. Around 2800 BCE, pastoral people from the Pontic-Caspian steppe, the Yamnaya, expanded westward into Central Europe, generating what archaeologists call the corded ware culture. Stolaric's team in Poland confirmed what Hack's earlier work established. The Yamnaya expansion replaced the Y chromosomes of the earlier farming population in Poland almost completely. Steppe ancestry now accounts for roughly 40 to 50% of modern Polish genomes. The haplogroups R1a and R1b arrive with this wave. This was replacement event number 1, thousands of years before any Slav, any Goth, any Roman had ever heard of the Vistula. Floor 3, the surface layer, the one that feels most Polish because it's the most recent. So Milewski and Dawidziuk at the Institute of Mother and Child in Poland analyzed 598 unrelated Polish male genomes in 2025. They found that nearly 97% of all R1a samples in modern Poland belong to just 2 sub-haplogroups, R-Z280 and R-M458, both confirmed as the core Slavic migration lineage, originating in the Dniester-Don corridor of what is now southern Belarus and central Ukraine. The Slavic surface is, in the male line, almost total. 3 floors. Hunter-gatherer base. Yamnaya steppe second layer. Slavic eastern surface. Clean, resolved. You can stop wondering now. Except, the Milewski and Dawidziuk 2025 study contains a finding that has received almost no popular press attention at all. Among the 23 Polish-specific Y chromosome subclades they identified, the 2, I-Y6343 and R-Z17913, don't fit cleanly into any of those 3 layers. The paper's own words describe them as, and I'm paraphrasing directly here, remnants of the local Iron Age population who survived the Great Migrations and then moderately expanded within the Slavic medieval community. What they found in those 2 subclades changed everything the previous generation of researchers thought about pre-Slavic survival, a pre-Slavic Iron Age survivor subclade, expanding quietly inside the medieval Slavic community. That is not a rounding error. That is a story. A small group of men who survived the Gothic departure, survived the Slavic arrival, and whose sons married into the New World and kept going. They didn't disappear. They camouflaged. And their descendants are walking around Poland right now, most of them with no idea what their Y chromosome is actually recording. But the number of them, and whether they represent a real population survival or a statistical artifact, that's where the science breaks open completely. Because this paper was published in October 2025, as of this recording, it has been cited fewer than 15 times. The most significant finding about Polish genetic uniqueness in a decade is sitting in a journal that almost nobody has read. Why? Why? Because the story those survivor subclades are telling is not comfortable. I-Y6343 and R-Z17913 are not just rare haplogroups sitting quietly in the data. Milewski and Dawidzic's 2025 paper dates their most recent common ancestor, using mutation rate calculations, to a founding event consistent with the 5th to 6th century CE window. He is right in the middle of the migration period, right in the exact moment when the male gene pool of the Polish Vistula corridor compressed to near zero. This discovery was published in October 2025. Nobody has written the headline. What this means is that I-Y6343 doesn't descend from a large surviving Wielbark population. It descends from a very small number of men who passed through something severe. Population genetics has a specific term for this: founder effect following near extinction. The kind of genomic compression you see when a population passes through a crisis so extreme that only a fraction of men survive to have children. The genetic signal contracts, a bottleneck. And on the other side of the bottleneck, only a few lineages remain. No war memorial marks this event. No archaeological site has confirmed systematic large-scale violence at the sites where the male lineage vanishes. What the record shows is quieter and, in its own way, more disturbing. Settlements occupied continuously for 400 years simply go silent. Cemeteries stop receiving new burials. The entire pattern of farmsteads and community organization across the Vistula corridor just stops within roughly 100 years, and the cause is unconfirmed: famine, disease, war, men voluntarily following the Gothic road south. The data records the compression. It does not record the cause. There is no agreed-upon name for what happened in that century in the scientific literature. The men who carry I-Y6343 today in Poland are the biological descendants, 40 generations removed, of the handful of Iron Age males who survived whatever that was. Not the Wielbark culture as a whole, not a named ethnicity. The biological heirs of a catastrophe that has no monument, no confirmed date, and no name. That is not a haplogroup designation. That is a scar inside a scar. And if you are a Polish man who has taken a Y chromosome test and found I-Y6343 or R-Z17913 in your results, you are not descended from the Slavic migrants who built medieval Poland. You are descended from whoever stayed, whoever survived, whatever that required of them. Now, here's where the ground really shifts. Stolarek's 2023 study found that the Wielbark genetic signal was indispensable for modeling the medieval Slavic population mathematically. And the survivors didn't just persist. They contributed. They were absorbed. Which means the Slavic gene pool that built modern Poland already contained a pre-Slavic layer when it arrived. The replacement was never 100%, and that small percentage changes the story of Polish genetic uniqueness entirely. But here's the problem. Two of the most authoritative studies on this question, published the same year by different research teams, directly contradict each other on exactly this point, and only one of them is being cited. The citation gap is not an accident. Two peer-reviewed studies, both published in 2025, both using ancient and modern Polish genomic data, both arriving at conclusions about pre-Slavic survival in Poland, and they do not agree. And yet, the data from the less cited study says the exact opposite of what the dominant narrative has been repeating. Here's the first study, Gretzinger and Krauss at the Max Planck Institute in Germany, published in Nature, the highest prestige scientific journal on the planet. 555 ancient genomes, more than 80% population replacement in Poland during the Slavic migration period. Pre-Slavic Y chromosome contribution to modern Poles, minimal, effectively non-significant at the population level. As of this recording, that paper has over 180 citations. It is the study everyone references when they talk about Polish ancestry. Here's the second study, Milewski and Dawidziuk at the Institute of Mother and Child in Poland, published in Human Genetics, a respected Springer Nature journal. She deep sub-clade analysis of 598 modern Polish male genomes, identification of I-Y6343 and R-Z17913 as Polish-specific survivor subclades with pre-Slavic Iron Age founders. The pre-Slavic male contribution is small, but it is real. It is specific. It has names. As of this recording, that paper has fewer than 15 citations, 180 versus 15. And Polish national ancestry hangs in the difference. The two findings are not easily reconciled. Either the pre-Slavic contribution is negligible at the population level, the nature position, or it is real, specific, and traceable to named survivors, the Milewski position. The conflict isn't purely about methodology. It's about something subtler, what you use as your pre-Slavic reference panel. And in ancient genomics, that choice is never fully neutral. When you define what counts as pre-Slavic, you are already making a decision about Polish identity. The reference panel is political, not because anyone intends it to be, because history always is. And there's one more thing in the Milewski and Dawidziuk paper that nobody in English-language science journalism has mentioned yet. Their analysis identifies Baltic-associated subclades N1A-Z16975 and N1A-Z16980, present in modern Poland and likely representing a contribution from Baltic fringe populations that predates the Slavic migration entirely. The Baltic thread in Polish maternal DNA that the 2014 Roble study hinted at, it shows up in the paternal data too. A separate Baltic layer, unnamed in any popular account, present, real. Scientific consensus is infrastructure, or an infrastructure can be built on partial foundations. The partial foundation being built around Polish ancestry right now leaves out the pre-Slavic survivors, the Baltic layer, and the women of Kołalewko, all in the same editorial sweep. Not because the data isn't there, because the data isn't getting cited. This 4,000-year demographic story, Yamnaya over farmers, Goths over Iron Age communities, Slavs over Wielbark remnants, is not ancient history being quietly resolved in academic journals. It is happening right now in citation counts and reference panel choices, and which findings get press releases and which ones sit in supplementary tables. And there is one final finding, buried in the 2014 maternal haplotype sharing analysis that connects modern Poland to a living population that has nothing to do with Slavic ancestry. And nobody has made this connection in public. Nobody. Because the connection points somewhere unexpected. When population geneticists plot modern Polish individuals on a genetic map of Europe, something specific happens. The Polish cluster is unusually tight, more internally coherent than Germans, more homogeneous than Czechs, more compressed than almost any other Central European population sitting at a major migration crossroads. That level of clustering is not normal for a group positioned at the intersection of so many historical movements. It means a recent shared founder population, a group that arrived together, stayed together, and didn't mix outward for long enough to compress. That compression comes from two events stacked on top of each other. The first was the Slavic migration itself, a relatively homogeneous founding population arriving from the east and forming the genetic base of medieval Poland. The second was the 20th century, the forced resettlements, deportations, and murders of 1939 to 1945 killed or expelled a massive portion of Poland's internal genetic diversity, Jewish communities, German-speaking populations, regional minorities. The Poland that emerged from World War II was ethnically sorted in a way no natural historical process produces. The tight, modern Polish genetic cluster is, in part, a direct consequence of the Holocaust. That is not a rhetorical point. That is what the genomic data records. Now, here is the parallel that no one is talking about. On the other side of the planet, the Japanese archipelago holds a story that mirrors the Polish one with an almost unsettling precision. The Yayoi people arrived from the Asian mainland roughly 2,000 years ago and replaced approximately 75 to 90% of the earlier Jomon population within a few centuries. The speed and scale are nearly identical to the Slavic replacement in Poland. Like the Wielbark remnants in Poland, the Jomon left detectable genomic traces in a small percentage of modern Japanese. Like Polish identity, Japanese national identity is built around the story of the arriving people, not the ones who were there before. In both cases, the founding mythology belongs to the migrants. In both cases, the oldest inhabitants of the land are genomically nearly invisible. The pattern is not Polish. The pattern is human. And here is the thing about modern Poles that their DNA test results keep showing them, and that keeps surprising them. Modern Poles cluster most closely on genome-wide comparisons with Ukrainians, Belarusians, and Baltic populations. Not with Central Europeans in the conventional sense, not with Germans or Czechs or Austrians. The people they feel most genetically connected to are to their east and northeast, because that is where the Slavic founders came from. Modern Poles are not the genetic heirs of the ancient Central European civilization that built Kołowko. They are the descendants of people from the East, who came, stayed, survived, and became something new. They are someone else's ancient diaspora, arrived, rooted, and slowly convinced that they were always there. Which brings us back to the question this entire video has been circling. If Polish identity is genetically recent, if the pre-Slavic population is genomically almost gone, if the Baltic layer is real but unnamed, if the survivor sub-clades are present but undercited, then what exactly are Poles inheriting when they invoke their ancient bloodline? The answer is not what the Warsaw Classroom said, but there is one more finding, published quietly, in supplementary data, in the paper that nobody cited, and it changes the frame of the entire story. The Vistula River is 1,047 kilometers long. It has been flowing through what is now Poland since before the word Poland existed. Every population in this video has lived on its banks, crossed it, buried their dead near it, or been replaced by someone who did. It doesn't remember any of this. But in the 2014 study, buried in a supplementary haplotype sharing table that almost no one has discussed in public, there is a number. If you're finding these questions, the ones that sit underneath the official story, the ones the data raises and no textbook answers, subscribe. The next video pulls this thread in a direction that will make the Polish story look simple by comparison. The Roble 2014 study tested haplotype sharing probabilities between Roman Iron Age Polish samples and every modern European population in their reference panel. The Roman-era Wielbark samples, the women specifically, shared rare mitochondrial haplotypes not only with modern Poles. They shared them at statistically comparable rates with modern Lithuanians and Latvians. The Baltic connection that showed up in the strontium data at Kołowko, the Baltic connection that showed up in Milewski and Dawidziuk's paternal subclade analysis. It appears again here in the maternal record, pointing northeast. The women along the Vistula who survived the Gothic departure and the Slavic arrival were not only Proto-Poles. In their genetic makeup, they were Baltic adjacent, closer on certain measures to the people of modern Lithuania than to the Slavic migrants who would become the fathers of modern Poland. Which means the oldest living genetic thread in Polish ancestry is not Slavic, it is Baltic. The founding mothers of Poland were not Polish. They were never asked. There is a specific mitochondrial haplotype, cataloged in the 2014 study, sequenced from a Roman-era woman at a Przeworsk culture site near Krakow, that matches a haplotype found in a living woman in Riga, Latvia, identified in a population survey in 2009. The two women share a maternal ancestor. The connection spans 1,800 years, two national identities, two languages, two entirely separate histories that have nothing to do with each other in any classroom in either country. The data does not care about that. The river flows north. It has always flowed north, somewhere in the sediment between the Wielbark graves and the Slavic settlements and the Yemnaya steppe echo and the unnamed Baltic women and the survivor subclades that a paper with 15 citations is trying to tell us about, somewhere in all of that, is the actual answer to who Poles are. Not what the whiteboard says, not what the citation count says, what the bones say, and the bones are still talking.